Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and what we were discussing about the isolation as well as the purification of the overexpressed product from the cells or the host. In this context, so far we have discussed about the two properties of the protein which can be exploited in two different techniques. In one of the as lectures, we have discussed about how you can exploit the presence of charge on the protein and in the techniques called ion exchange chromatography, and in the next lecture we have discussion about the presence of hydrophobic patches present on the protein surfaces and how that can be exploited to purify the protein using the hydrophobic interaction chromatography. Now, let us move on to the third property, and the third property is that the proteins are having the surface area or the surface area which can be utilized to purify as well as to characterize a particular protein, and the technique which is related to the surface area is known as the gel filtration chromatography. Now, before going into the details of the gel filtration chromatography, let us discuss how we can exploit the surface area of a protein to characterize the particular protein as well as how that can be used to purify the different proteins from a complex mixture. So, now you can imagine that proteins are nothing but the made up of a peptide chain which is made up of different amino acid residues. So, initially when they are being synthesized from the ribosome, they are being synthesized in the linear chain, and that linear chain form is called as the extended form. So, when they are being synthesized from the ribosome, they are synthesized in the extended form. As soon as the chain is coming out from the ribosome, it is based on the microenvironment, then it starts folding by the interaction between the residues. Like if the residue is present here, it can make an interaction with the neighboring residue and so on, and ultimately you are going to have the partially folded protein, and then that partially folded protein is going to be get converted into the fully folded protein. And what you can see in a fully folded protein, that the hydrophobic patches are going to be present in the center, whereas the hydrophilic patches hydrophilic molecules are going to be present on the surface. And by doing this whole process, what protein is trying to do is it trying to conserve the free energy, and it is trying to reduce the surface area, and because of that it is actually trying to make the structure as compact as possible. That you can understand by this diagram. So, if you see a protein from the top, what you will see that there is a center. Around this center the proteins are arranging its amino acid residues, and as a result the protein is going to form a globular structure. And in this process what it does and how this actually arranged the amino acid is based on the environment which is present outside, which means the local environment. If you have and most of the cases the environment like the pH, the salt and all other factors which are present in the microenvironment are actually going to induce, or it is going to affect the packaging of these protein molecules in such a way, but at the end it is going to arrange along with the center. And that is why if you would like to characterize a protein, you can actually characterize simply by knowing the radius of gyration or the diameter of that particular ball. And what you can see in a fully folded protein, that the protein will arrange the hydrophobic molecules in the center and the hydrophilic molecule around it, and that is required because the microenvironment in most of the protein cases is hydrophilic in nature, which means that you have a water outside, and because of that all the polar as well as the charged residues are being arranged outside, and all the hydrophobic amino acids which does not like the water or the aqueous environment are getting focused into the center of the protein. And by doing this process of folding a protein around a particular center, the protein is trying to conserve the energy as well as it is trying to reduce or make the structure as compact as possible. And in this process what happen is most of the globular protein are making the ball like structures, and if you compare these balls you could be able to characterize these proteins as well as you could be able to purify. Let us understand this in a more elaborated example. So, now we have synthesized a protein, and what you see here is I have given an example of the protein which are of different molecular weight. So, it starts from the 5 kilodalton, 15 kilodalton, 35 kilodalton, 65 kilodalton and 95 kilodalton. What I have done is I have shown you the number of amino acid residues which are associated with most of these proteins. So, what you see is for the 5 kDa protein you have the number of residues which are 45, for 15 kDa it is 135 and 35 it is 315 and so on, and these are the rough estimates. And now based on this number of residues I have also calculated the size of these or radius of gyration of these proteins. So, what you see is that the protein which is of the 5 kilodalton is having a radius of gyration of 2.545 nanometer, whereas in the case of 15 kilodalton it is a 3.53 and 4.69. So, what you see here is that as you are increasing the size of the protein, it actually increasing its radius of gyration, which means as you go from a smaller protein to a larger protein, and all this is applicable only for the globular protein, that the diameter of that particular ball is increasing, which means if you consider that only the globular proteins are present in a complex mixture, you are actually having nothing but a combination of different balls of different sizes, which means you have a protein of different sizes or different balls are present in the microenvironment, and all these balls can be separated simply by reducing or simply by filtering these protein particles through a different pore sizes, which means if you in a general term what you can do is if you want to separate a protein of 5 kilodalton from the 65 kilodalton. So, you can see that 65 kilodalton is of this diameter compared to the 5 kilodalton which is of this diameter, and if you have a mixture of this protein and this protein, what you can do is you can simply take the filters of two different diameter, the diameter which is corresponding to the 5.77 nanometer and the diameter which is corresponding to the 2.45 nanometer, and if you use these two filters and if you filter this protein and this protein, what will happen is these two proteins are going to be separated from each other, and this is what exactly the gel filtration is going to do. It is actually having the funnels of different pore sizes, and based on that it is actually filtering these molecules. So, in a typical gel filtration what you have is you have the beads which are having the large pore size, you have the beads which are of smaller pore size, and in general when you put the molecules through this, the large size molecules are going to be excluded, whereas the small molecules are going to be included. This is actually exactly the reverse. In most of the cases when you are doing a filtration, the small molecules are getting passed through the diameter, whereas the larger molecules are going to be retained. Whereas in the case of gel filtration chromatography, you are actually going to exclude the larger molecules and you are going to retain the smaller molecule into those pores. That is why the gel filtration chromatography is also called as the reverse sieving techniques, or it does not follow the filtration principle. In the normal filtration principle, what you have is, suppose I am using a filter of 10 nanometer. So, what that mean is that the particles which are lower to this are going to be filtered out, whereas the particles which are of larger size to this are going to be retained onto the filter. Whereas in this case, if you have a bead of 10 nanometer, which means you are going to exclude or you are going to reject all the molecules which are actually of larger size to 10 nanometer, whereas you are going to retain all the molecules which are of the lower size. That is why the gel filtration chromatography actually follows the reverse principle of sieving. Now, let us understand how the gel filtration chromatography works. So, in this particular chromatography, the column is packed with the beads containing the pores of different sizes, and that actually is going to resolve or going to allow the entry of molecule based on their sizes. So, you can imagine that you have injected the a complex mixture of the protein molecules which are of different diameters of like orange, green, red and so on. So, all these molecules are going to distribute within these beads, and once they distribute within these beads, because these beads are not the normal beads, these beads are going to have the pores. So, they will actually going to distribute between the space which is present inside the pore and the space which is present outside the pore. So, if the molecule is of a smaller diameter, it will get filled into those pores, whereas the molecule which is of larger size is going to be excluded from these pores. So, a smallest size in the inner part of the pore, followed by the gradually increasing size and the largest molecule excluded from entering into the gel. This means here also the molecules are going to be distributed from the outside buffer into the buffer which is present inside the pores. So, it will be get exchanged with the buffer which is present inside the pores, and the smallest molecule is going to sit at the bottom of the pore, whereas the larger molecules or more larger molecules are going to sit on top of the small molecules, and the largest molecule which will not enter into the pore is going to be excluded from these pores, and that will remain into the buffer phase. The separation between the molecule occurs due to the time they travel to come out from the pore. When the mobile phase passes through the column, it takes protein along with the smaller molecule present in the inner part of the gel takes longer time to come out and travel longer path to come out, whereas the larger molecule travel less distance to come out. As a result, large molecule and a small molecule get separated from each other. Now, let us understand, so what is the kind of pore is present and how the separation is occurring in a gel filtration chromatography. So, if I show you a pore, this is the gel filtration bead, okay, and the pore is present in a funnel shaped pore which is present on these beads. So, you can imagine that you have a funnel shaped beads which has a pore like this from all the sides. Okay. Now, what will happen is, while the molecules are flowing on top of these beads, they will going to enter, they will going to replace the buffer what you are filling into these beads, and they are going to fill. So, the smallest molecule are going to sit onto the bottom of these beads, whereas the molecule which is larger to this is going to sit on top of this, and the molecule larger to this is going to sit on top of this, and the largest molecule is going to sit on top of this, and the molecule which is even larger to this is not going to enter into the pore, and it will go away along with the buffers. Now, this is the situation when you have initially injected the molecule and they are actually passing through the different layers. So, this kind of distribution is going to occur from the first layer to the last layer of the column, which means the first layer is going to have the smallest beads, smallest objects. The second layer is going to have the objects which are larger to this, and the third layer is going to have the objects larger to this. So, if you understand or if you have a confusion that the every bead is going to have the molecules of these molecules arranged in a pores, then that is not true, because the dynamics of these molecules are very different. Because of their sizes, the dynamics of the small molecule is going to be very large. That is why when the molecules are going to be distributed from the buffer which is filled within the pore versus the aqueous environment which is present outside, in the first layer when that distribution occurs, the objects are going to be filled at the bottom of the pore, and by the time the next molecule will try to fill into the same pore where the already existing molecules are present, it will get over and flow by the liquid, and then it will be getting exchanged into the next layer, and in the next layer it is going to sit like this, and then the third layer it is going to have this, and ultimately the last layer is where the molecules are going to be excluded because they will not enter, and that is why they will come out from the aqueous phase and they will going to come out in the from the column, and they will be present in the flow through. So, that exchange occurs at every layer, and as a result throughout the column you are going to have the molecules filled within these beads, and that is why you can imagine that this molecule has to travel all the way, this molecule which is filled in the first layer has to travel all the way from first of the column to the last of the column, and that is why the smaller molecules are going to travel larger distances or larger volume, whereas the larger molecules which are being present within the beads which are very far away from the smaller object will going to travel smaller distances, and that is why the large molecules comes first and the small molecule comes later, and the largest molecule which will not enter into the beads are probably present somewhere here, and that is why they will come out very early on, and they may come out within the wide volume. Now, suppose you are preparing a column and the total volume of the gel what you have used is called Vt. Then the Vt, which is the total volume of the column, is equivalent to the Vg plus Vi plus Vo. What is mean by the Vg is the volume of the gel matrix, which means the volume of these beads. Vi is the pore volume, which means the volume of these pores which you are going to which is going to be taking part into the exchange, and Vo is the void volume, which is actually the volume which is being excluded. So, void volume means the volume which is being avoided by the molecules and which will not take parts into the exchange or the distribution phenomena. The volume of the mobile phase flow to elute a substance from a column is known as the elution volume or the VE. The elution volume is related to the void volume and the distribution coefficient Kd. So, the elution volume VE is equivalent to VO plus Kd Vi. Okay. So, if you rearrange, the Kd is equivalent to VE minus VO by Vi. So, that is the formula for Kd. Okay. So, Kd is the ratio of inner volume available for an analyte is the independent to the column geometry or the length. As per the relationship, 3 different types of analytes are possible in a gel filtration column. Now, imagine that you have an analyte which has the Kd is equivalent to 0. Kd is 0. If the Kd is 0, if you put the 0, okay, then what will happen is that VE minus VO divided by VI. So, if you do if you do the rearrangement, what you will see is that VE is equivalent to VO, which means that VE is equivalent to VO means these analytes will be completely excluded from the column. So, if the Kd is 0, which means the molecule is not entering into the pores of these beads, and because of that the Vi is going to be 0, and as a result the VE is going to be equivalent to VO, which is wide volume. Now, you can imagine that you have an analyte which is having the Kd is equivalent to 1. So, if the Kd is 1, then it is VE minus VO divided by Vi, and if it is Ve, so Ve is equivalent to Vi plus Vo, okay, which means the Ve is equivalent to Vo plus Vi, which means these analytes will be completely in the pore of the beads, which means if you see the beads, the molecule which are sitting at the innermost area of the beads, at the innermost of the area of the beads is going to give you a Kd value which is 1. Now, if you have a Kd value which is actually the Kd is bigger than 1, which means now you can imagine that if the Kd is bigger than this, if the Kd is bigger than 1, that what that mean is that the Ve is going to be bigger than Vi plus Vo, okay, which means in those cases the and in what conditions the Ve is going to be bigger than Vi plus Vo, under one condition that the molecule which you are resolving is irreversibly binding to the beads. So, one of the things which you have to consider while you are doing the gel filtration is that while you are doing the gel filtration, the molecules are only getting exchanged between the pores versus outside area. They are not getting associated with the beads either by the non-coordinate bonds or the coordinate bonds or any kind of interaction, which means if you even if your protein has a charge, the beads are not going to have the association with these molecules because there is a charge present. So, that is why most of the beads what people use in the gel filtration does not have any kind of affinity or any kind of preferences or any kind of association with the molecules. But if the molecule is going to show you the Kd which is bigger than 1, which means the molecule has irreversibly been bound to these beads, and in those cases you can flow the liquids, but the molecule will not going to come out, which means what I am saying is if the molecule will go and bind to the one of the pores, then even if you flow the liquids, because we are not going to do any salt gradient or any kind of such kind of treatment, that is why the molecule will remain stuck to these beads and it will not come out. So, that is why if the Kd is bigger than 1, in this situation analyte will adsorb onto the column matrix, and if that happens, then you have to wash this column, you have to strip this particular molecule from the beads, and then you have to repack, which means if that happens you have to repack the column and it is going to destroy the column packing. So, this is what we are talking about. If you resolve the molecules of different sizes, if you inject the molecule, what you will see is the large size molecule will come out first, the medium size molecule will come out later, and the small size molecule or the smallest size of molecule will come later, which means the elution volume for the large is going to be very small, the elution volume for the medium is going to be in between, and the elution volume for the smallest one is going to be largest, and that is simply because the small molecule has to travel from all the way from the top of the column to the bottom, whereas the medium one has to travel the medium distance and the largest one has to travel the smallest distance. That is why they will come out on a different volumes. Now, let us see what are the metrics you can use for gel filtration. The choice of column depends on the range of molecular weight and the pressure limit of the operating equipment. If you remember, when we were discussing about the protein purification system, we have discussed about the different types of pumps, what you can use. You can use the low pressure pumps, you can use middle pressure pump, and you can use the high pressure pumps. Similarly, you can also use the molecular weight. So, you should always know what molecular weight is my target molecule. So, that is why you can use the gel filtration chromatography matrix based on the pressure limit which pressure limit which you are going to use, and as well as the molecular weight range which you are going to resolve. For example, if you use the Cefadex G10, the G10 is a matrix which actually can fractionate from small molecule to 700 Dalton, whereas in the case of Cefadex GT100, so you can see that it actually can resolve from 4 kDa to 150 kDa. So, what is mean by the fractionation range? The fractionation range means this molecule will be the largest size which this column can resolve, and this is the smallest molecule which is this molecule can resolve. So, that is what is the fraction that decides the fractionation range, which means within this fractionation range the column is going to have the better resolution and it is going to fractionate the different molecules. But if you take the molecules which are larger to this, for example, if you take a molecule of 180 kilodalton, that does not mean that 180 kilodalton will not going to be resolved, but if you see a separation between 150 versus 180, these two molecules will come together compared to if you are resolving the 120 and 150. The difference between 120 and 150 is also 30 kilodalton, the difference between 150 and 180 is also 30 kilodalton, but because the column has a fractionation range between the 4 kDa to 150 kDa, it will going to resolve 120 kDa to 150 kDa better compared to the from 150 kDa to 180 kDa, because the pore size will not allow the 180 kDa to be resolved from the 150 kDa. So, the both molecule will sit on top of each other, and when you elute, they both are going to come together. Now, let us see how to do the operation of the gel filtration chromatography. So, one of the crucial parameter of the gel filtration chromatography is that the column packing. So, the first event is that you have to do a column packing. So, in the column packing of a gel filtration chromatography, you have to be very careful. For example, the column material what you are going to get from the companies are either be present in a liquid phase, or they will probably give you the column in a powder. So, if you are getting a column in the form of powder, then this powder has to be put it into the buffer, and then you have to put it in these buffers into the buffer, so that the column will get swell. And so, if the column will get swell, it actually going to increase its volume, and that is how you have to use this particular column. If it is present in the liquid form, then mostly the column material is always been stored in 20% alcohol containing the 0.2% azide. In those cases, then you have to do at least 3 to 4 time buffer exchange, so that there will be no alcohol present in that column. Now, once your beads are ready, either they are being swollen from the powder, or you have already got the swollen beads from the companies and you have washed and prepared it, you have to allow the column material to settle down in a mobile phase. So, mostly you have a glass column which actually has a filter at the bottom, and you have a plunger which you can use to pack the column. So, through this plunger you are going to pour the material. So, it is going to pour into the glass tube and allow the beads to settle without trapping the air within the column. So, while you are settling down, you are also going to flow the water or flow the buffer through a pump. So, you can use the peristaltic pump, or you can use the pump which are present in the protein purification system. Now, you suppose, so what will be the relationship between the column packaging flow rate versus the flow rate at which you can operate this column? Now, suppose I am packing this column at 5 ml per minute. Okay. Now, if I am packaging this column at 5 ml per minute, it is recommended that you should not use this column beyond 1 ml per minute. Why it is so? Because if you run it beyond 1 ml per minute, you probably going to affect the column packaging, because if you remember, the gel filtration is where the beads are arranged in the column, and from the bead number layer number 1, the molecules are going to start fractionating. In the bead number 2, the medium size molecules are going to fractionate, and that is how. So, if you change the packaging, if you change the amount of interspatial or interbead spaces, you are going to either decrease or increase the number of layers, and that actually is going to affect the overall fractionation of the molecules within that particular column. The other thing is that you should always try to avoid the bubble or any air bubble or any kind of air within the column, and that is very important for the chromatography of gel filtration. That is very important if you would like to resolve the molecule on a gel filtration chromatography column. Now, once the matrix is settled down to give a column, it can be tested for presence of air channels and well packing by a flowing analyte with Kd equal to 1. It is expected that the elution volume in this case should be VO equal to Vi. So, now apart from the air bubble, you also have another problem of air channels. What is mean by air channel is, suppose you have packed a column and it is continuous layer of beads from bottom to top, and in between some layer you have a channel of air. Okay. So, how the channel of air is going to disturb the overall purification? What will happen is, as soon as the molecule will reach to this particular channel, they are not going to fractionate from the pores to the aqueous environment, because they have a free movement of within this channel. So, they will travel within this movement within this channel, and they will not going to fractionate between the buffer what you are using versus the pores which are present on the beads, and because of that they will going to mix up with the molecules which are already present in this particular layer, and as a result they will not going to follow the principle of gel filtration chromatography, which means they are not going to be separated nicely, and they will get mixed up with the other molecule, and as a result it is overall it is going to affect the separation or the resolution of this particular column. Now, the question is how you can test that there is no air channels. Now, if you would like to test that, you have to pass through a material which is of Kd equal to 1, and if you remember, we have said that what is mean by Kd equal to 1 is the molecule which is going to sit at the bottom of that particular pore, and that should take the amount of time which is actually equivalent to VO equal to VO plus Vi, which means it should travel all the way from the pores which is actually the Vi, versus it should travel all the way from the top to bottom. What could this molecule could be? Okay, the simplest molecule could be the water molecule, because water molecule is the simplest and the smallest molecule which can be present on the bottom of these pores. But the problem with the water molecule is that water molecule you cannot monitor until it is radio labeled, and we are not going to do a radio labeled experiments just simply by knowing whether there is a channel or whether there is a air bubble present in this column or not. So, to know this particular aspect, you can use some molecule which is of smallest in nature, but it should be getting detectable. One of such molecule is the acetone. Acetone is a organic molecule. So, its immiscibility within the water is less, but it still can be a very small molecules. So, the acetone will sit at the bottom of this particular well, and acetone is compared to the water, acetone absorbs very strongly at the 220 nanometer, which is actually absorbance for the most of the aliphatic compounds, the compound which contains the carbon-carbon bonds, and you can use acetone. So, what you do is you layer the acetone. So, acetone will get filled at the bottom of this particular pore, and then it will travel all the way. So, once the acetone comes, it will give you a peak in 220, and by knowing the peak, you could be able to calculate the elution volume, and if the elution volume is equivalent to the Vi plus Vo, which means you will know that actually the column does not have any channels, column does not have the pores. That does not mean that it is not going to develop the channel or the pores, air bubbles. That for that only the most of the gel filtration column has to be packed in the same temperature where you are planning to operate these columns, which means if you change the temperature, if you change the temperature of packaging from the running, you know the temperature is going to make the expansion of the liquids, or it is actually going to expand the air which is present within that particular liquid. And if you change the temperature, suppose you go from 4 degree to 37 degree, then you are actually expanding the air which is present in the water in which your column is being packed, and because of that this particular expansion of this air, which is water soluble air, is going to come out, and it is actually going to create the channel as well as it is going to create the bubbles. Other than that, if you would like to avoid the air bubbles or the channels, you also have to use the water which is degassed. What is mean by degassed is that you can reduce the amount of dissolved oxygen, and by degassing you can do simply by putting that particular liquid into the vacuum, or you can also do the sonication. Both are these processes are actually removing the oxygen from the water. Now, once the packaging is over, then you can load the column and you can prepare the sample in your mobile phase. So, whatever the condition of your mobile phase you can use, and you have to ensure that it is free of suspended particles, so that the column is not going to be clogged. The most recommended method to apply the sample is to inject the sample with a syringe. Now, once you are done, you can do the elution. So, compared to the ion exchange chromatography or the hydrophobic interaction chromatography, in gel filtration there is no gradient of salt to used to elute the sample from the column. The flow of mobile phase is used to elute the molecule from the column. Once you are done with the elution, you have to do a regeneration. So, regeneration, after the analysis of the analyte, gel filtration column is washed with the salt containing mobile phase to remove all non-specifically adsorbed protein to the matrix. The column is then equilibrated with the mobile phase to regenerate the column. The column can be stored at 4 degree in the presence of 20% alcohol containing 0.05% sodium azide. And just like as we discussed for the ion exchange chromatography or hydrophobic interaction chromatography, the column is always being stored in 20% alcohol containing 0.05% azide, so that you will not going to see a growth of bacteria. So, with this I would like to conclude our lecture here. Thank you.